Good evening. This is the Advanced Science Academy at Morris Catholic High School. I wanted to give you a brief update before Mikey starts. We have started putting your fees to good use. I have presently, actually over in my living room, I have, because for some reason they delivered it to me, I have a brand new $7,000 centrifuge that I got for $500 on eBay. I'm going to plug it in tomorrow and it's not going to work. And then I'm going to be like, well, we're out $500, but it's warranted for a year. So, or 2 years, something like that. So I think that it should work. It's not pretty, but it's the one that most labs I worked in has. I'm going to try to get us a PCR machine. I got us some electrophysiology equipment. So we'll learn how to use that during the next neuro session, either AP or when I teach neuro 2 years from now. Then we also we have officially ordered the best resin 3D printer you can get. It's called a Peopoly L, a Peopoly Phenom Model L. So it's the large format one. 3D prints in resin using an LCD screen, which is pretty cool. I'm also trying to order, but they're not taking my order for some reason, a brand new extrusion 3D printer, and that's also the best one you can buy based on the reviews. And that one is a Prusa M3KS, I think is the model. We also bought a whole bunch of dissectable stuff. We bought some dissection materials. We bought some chem models. So we're starting to beef up the science department. It's not going to resemble anything you've seen since you've been in Morris Catholic. Right. Next week is going to be Dr. Ashley Bassett, who is an orthopedic surgeon. I need to send her an email just to make sure everything's good to go. She said she was good to go, but it's just appropriate professional to make sure they touch base. And then after her, I believe will be Chin will be giving the next journal club. And we do have fast approaching our first dark sky event in April. That is scheduled for the first week we're back in school. However, it was scheduled not knowing that we planned on not going into school that week. So I need to check with Mr. Cleffy and see if we're allowed to do that. We may not be able to. I might have to jigger the schedule to see where else we could do that. The reason it was that date was that's the date of new moon. So that's when the vampires come out. I mean, that's when there's no extra light, so you can see things with a telescope. See, my pop culture references have already expired from Twilight. I bet none of you have actually read Twilight, huh? You don't read books anymore, this generation. Like, I've seen the movie. I saw one of the movies. That's probably what their answer would be. All right, let's get the show on the road. Mikey, tell us why cats are the most evil demons on the planet.
So the journal that I'm presenting is about the susceptibility of certain domesticated animals to SARS-CoV-2, which is a strain of COVID. So it's a way of seeing the susceptibility of certain animals to COVID, which is really important right now, obviously, because of what's going on. So first they conducted the trying to think of the word. they basically they gave ferrets SARS-CoV-2, and they saw where it replicated most in the ferret systems, and A and B show that it replicated most in the nasal turbinate and soft palate and the tonsils. So it shows that it replicated the most in the upper respiratory tract of the ferrets, but like there was nothing in the rest of the body. So figures E through H, or E through I can't speak. So E through H, it shows how much of the virus was present in the ferret systems after X amount of days. And the majority of the viruses in the ferrets that the difference in so the virus usually stayed for 8 days after inoculation, but then it dropped off on the 10th. And then I through L shows how many antibodies were present in the ferret system for each of of 3 ferrets that they tested. And the ferrets with the asterisk were ferrets that had to be euthanized earlier. So that's why their antibody count is lower than the rest of the ferrets.
What was the reason for that?
It didn't really say in the journal, but I'm pretty sure it was due to complications.
Interesting. So we all need to, this is not just a Mikey thing, but all of us need to get in a better habit of actually orienting the reader, the people you're presenting to, orienting them to the axes and what we're reading. So in A and B, what is the y-axis?
The amount of viral RNA that was in the system of the ferrets.
Okay, per gram, right. So what's on the x-axis?
Where the viral RNA was found.
Okay, what's the dotted line?
That was where it was considered the normal amount of like viral RNA in the system.
Baseline background, yeah. So this is saying that the virus was replicating in those 3 areas: nasal, turbinates, soft palate, and tonsils?
Yes.
And that was confirmed in C and D?
Yes.
Okay. So again, now orient us to E through H.
E through H. E and F measures the amount of viral RNA that was in the system each day. And then G through H measures the amount of RNA, but through viral titer.
Yeah.
And that was for each day.
Okay. What is OD 450?
What?
What is OD 450? The y-axis for I through L.
That was the way that they measured like the amount of antibodies in the system, correct?
Mm-hmm. OD means optical density. So this is using some sort of high resolution imager that's measuring how much of a band you have. I'm surprised they didn't show the actual gels. Usually those are shown as a proof of principle in these type of papers, but they didn't. Okay, keep going.
Okay. So then figure 2, so after they performed the test on ferrets, they performed it on cats, and A and B they measure the amount of viral RNA that was in the system in each area of the cats, like in the ferrets, and they found a much wider range of where the RNA was able to replicate in all of the cats. And it was not just in juvenile cats, but also in subadult cats.
Now, is that because cats are demon spawn? Yes.
Maybe.
Cats are horrible, horrible things that, yeah. Okay. Just want to make that clear for everyone. Apple computers and cats. It's bad moves.
Followed closely by poodles.
You shut your mouth.
And then figure 3 is again with the cats, but this time it's measuring how much of the viral RNA was left in the system during each day. And it seemed to have dropped off after about after 5 days. For that's for the subadult cats, but then for the juvenile cats in E, it shows that the amount of viral RNA that was in the system seemed to have lasted longer, or yeah, replicated more in the juvenile cats than in the subadult cats. And C and F are again the amount of antibodies that were found in the cats after how many days, and C and F were measured through ELISA. So and then D
Eliza.
or ELISA, and then D and G were measured through neutralization assay. Was the amount of antibodies measured through neutralization assay. And again, the asterisk, it shows the cats that had to be euthanized before 20 days. And then figure 4 shows the info for other pets, and it shows that pigs, chickens, and ducks didn't seem to have any of the SARS-CoV-2 replicate in their system, but and dogs had less than ferrets and cats, but were still able to contract it. And then the conclusion is that ferrets and cats are highly susceptible to SARS-CoV-2. Dogs have low susceptibility, while other animals like ducks, chickens, and pigs are not susceptible. And in order to test vaccines, cats and ferrets should be used. And oh shoot, that's a typo.
Ferrets and cats are more likely to slap themselves in the face.
I forget what I was going to say there.
Get infected. Spread
yes. To spread infection to other people.
Okay. But do you think that result jives if you go back and read their abstract? Let's see. Boom. So here's their, for those of you who can pin me or whatever. Here's their abstract, and it says here that we found that SARS-CoV-2 replicates poorly in dogs, pigs, chickens, and ducks, putting dogs, pigs, chickens, and ducks kind of in the same category. Yet the data you showed, which is the table. So they didn't even make a figure for dogs. One virus inoculated beagle was euthanized on day 4. The virus was not detected. Right. So really you're only seeing it in rectal swabs, right? Am I reading that correctly?
Yes.
This is, Cam was saying this in my AP class, that this is how they're going to start testing humans for it, was rectal swabs, because they're more accurate. And at first I thought he was pulling our leg, our collective AP leg. And no, he was serious. He sent us the paper from China. But this would, because it's only rectally, it would not suggest then that they can really spread it. Right? Does that make sense? Unless you're eating their poop, which, I mean, who doesn't, right? Who doesn't see a nice fresh pile and be like, warmed up our, So okay. I want to shut up, and I want you guys to be critical here. Okay. We had some good papers to start. We had Catherine's paper, which she picked, which was a bit of the paper was a letdown. She did a good job with it, but I thought it was good that she chose a paper that was weak. Is she here? Let me see. Catherine, you here? Catherine, I don't see her. Well, what are the weaknesses that you see in this paper?
I thought that it was pretty weak that they didn't have any of the graphs for the other animals. So they didn't.
This is published in Science. It's one of the best. It's one of the big sexy 4, right? It is one of the best journals in the world. Science rarely publishes negative data. So they probably didn't include the graph because it didn't actually have any positive data in it. It wasn't an association. But yeah, I mean, why not put that in the same the way they displayed this? I thought was kind of odd. It was not an efficient use of space. That they probably could have done this entire paper in one figure. Miss Liv, you agree? You just combine everything into one figure, make it easy to read. But they spread it out. But they got a science paper and I didn't. So, who am I to talk, right? What else did you guys see?
Did they ever mention why they euthanize these animals early?
Mikey thinks there's a complication, right? Doesn't really say.
I think for the cats under.
Well, they're evil. We don't care about them. Right.
During the explanation for figure 3, I think it mentions that they had to be euthanized due to complications.
I feel like that would be an important.
Yeah, you'd think. What does that mean? Complications? What?
Yeah.
Because I can understand. I used to do brain surgery on mice and rats. We would occasionally lose some of them because the cannula fell out and they got infected, or they got some type of infection and died, or sometimes we didn't know why they died, but they died, and we'd have to make a note of that and try to at least talk about what went wrong, so that people who read the paper who try to do the same thing won't do the same thing wrong. That's why it's so important to have a methods section, right? So saying that they had to be euthanized is kind of a weird, I don't know. I wonder.
I mean, especially when they're literally measuring immunity. So if they're killing them off and not explaining why they have to,
Yeah.
really,
There are supplementary materials, and they might be in there. I'm just not sure. Hello? Not responding. Uh-oh, I think I froze it up. Hello. There might be some in there in the supplement, and I just didn't look at it. Request a pro. Oh, that's a request. I don't need that. Oh, that's nice. They actually provide the protocol. If you want to go get it, you can request the actual protocol. Interesting. So if I go to the website, here, I'll switch this over. If I'm going to the website, I go over to Journal Club. See, I try to provide all this stuff for you guys so you can do the same thing. Go to Mikey's section, go to the actual DOI, and then go to, where is it? Nope, I saw it a moment ago. Supplementary materials. Download supplement. Let's see if it's on there. Say F. F. Look for euthanize. Oh, euthan. 2 ferrets from each group were euthanized on day 4. Inoculations in the organs, tissues were collected. Didn't say why. Okay. Oh, here it is. The ferrets were scheduled to be euthanized on day 20 post-injection PI to assess them for antibodies. That's weird. They never finished the thought. Right? Am I right? Am I reading that right? It's like they were scheduled to be, and then they just like went off on a tangent about something else. You can tell this is also, it's very easy to tell that this was actually English is a second or third language here. It's not written by a native speaker. Usually Science and Nature provide editors that are fluent in the original language so that they can provide feedback and some suggestions on how to make it better. But you know, in the age of COVID, where if you have a COVID find that would be interesting, you try to pump it out as fast as possible, then that kind of doesn't get the same attention it would normally get. Yeah, this does not say. Let's see. Cats. Yep, nothing. No reason. For those of you who are wondering, like what breed of dog they use, they do tend to use beagles. Beagles are kind of the main dog breed that you would do research on if you're going to do research on dogs. I can't do that. I love dogs too much. Cats, I have no problem with the cats. Cats, no problem. The one time I had a bunch of mice that I needed to euthanize, and I walked into the necropsy, it's called, a place where, death happens. Sam, you'll be working there eventually with your love of trying to kill people. So I walked in and they had a dog. Two of the vets had a dog splayed completely open, and they were looking in its chest. And I went, "Whoa, sorry." And they're like, "No, come over here. We want your opinion." And I was like, "What am I going to possibly?" So I look inside, and they're like, "We're thinking that it was a heart attack." And I'm like, "Yep, sounds good." I have no idea. It's not my area of expertise. I was just like, "Yep, sounds good. Can't look at this dog anymore. Bye." Because puppies, right, kittens? Who could ever do that to something as beautiful as this little rat dog? Okay, onward. All right. Other problems you might have with the paper?
no one can see you or kittens. We can only see your paper on the screen, by the way.
Oh, I forgot. Other problems with the paper?
Why would they test ferrets? People actually own ferrets? Do you guys actually have a ferret?
I was just watching a video by a psychologist who had a ferret running around in the background. I thought that was weird too. But does anyone here have a ferret? Or a mink? Or a wolverine? Or a pet raccoon? Pet dolphin? Nothing? Okay. Come on, guys. This is awful. Help me out here. What's weird about this paper? There's like nothing bothers you about this paper? I am thinking specifically of a problem that one of you has raised in the past for other papers. Except this one actually does it wrong.
The end of the paragraphs don't summarize what the paragraph is talking about.
No, yeah, no. I don't mean in the execution of the writing. I actually mean in the science.
What's the implication of this finding?
Okay, that's a good question, Mikey.
This is going to be a really stupid question. But what would be the difference between an implication and like what the paper means?
They're the same.
Oh.
Implication might also include like why it's important to use specifically, as opposed to.
Okay.
Yeah, like to humanity. Why is this important?
Because it shows that if we were to for testing vaccines, it shows that ferrets and cats would be a lot better for testing vaccines than using other like domesticated animals like dogs. And yeah,
Why are they better?
because because it replicates better in ferrets and cats.
I think we're still unclear on why that's novel. Why is that interesting information? Do you guys know what PETA is?
Yeah.
People for the eating of tasty animals. People for the ethical treatment of animals. PETA. PETA forever has been running an ad campaign claiming that no animal has ever been infected by any human pathogen ever. That is a bold-faced lie, okay? Most pathogens that humans have are actually zoonotic in origin. They come from other animals. And our diseases and stuff can be, for the most part, transmittable to other, especially mammalian species. But you see it also in vertebrates. I mean, you're talking about chickens. Chickens are not mammals. They're dinosaurs. They're, reptilian, avian dinosaurs. So when you find that a species is more susceptible to a virus than another species, it makes it a better candidate to study the virus in, because they're not all going to react the same way. That's why it matters, right? Yeah. So, I mean, the implication that I thought was important was that all cats are evil, right? And that they're trying to kill us. I mean, they're okay with, they probably are the ones who infected all of us with coronavirus. Sam, you were probably playing with a cat while you were in California, and it was like,
I wasn't in California.
where were you? When Washington or Oregon? You were in one of those states, weren't you?
Arizona.
No, you weren't. You were in like the Northwest. You don't know what you're talking about.
I wasn't.
So cats are horrible. That's kind of what we're getting at. Yeah, cats. Anybody have cats? Who has cats? Hit the raise hand. Okay, so there's going to be a lot of euthanasia tonight, right? So I'll say a prayer for their souls. They'll probably go to hell anyway because they're cats, and then there you go. No, come on. Is there a problem with the study? I mean, I think the one about why they euthanize things, I hadn't actually noticed myself until you guys mentioned it. So I think that's a serious problem. I mean, this is a science paper. Science means you better have extraordinary results to get into one of the best journals in the world. And yet this sort of slips by. But there's a bigger problem with it. Come on. I'm expecting RC to say what it is because he said what it was last time. RC, pressure's on.
Is there no like comparison to like
With?
humans? Like if that's the
Oh,
implication, like
that
what's
would have been a good idea, wouldn't it? I didn't even think of that. Like, what if this is important for humans? Then why didn't they include somebody else's? You can do that. You can cite somebody else's data and say like this compares to such and such at this rate from a human. Good point. That's not what I'm talking about, but good point. That's a good point. So look, you guys are capable of doing this. You're capable of ripping apart a paper, finding the problems with it, and also hopefully admitting when it's good. You know, those first two we did were really, really phenomenal. So, well, not the weather one as much. That was okay. But the first one was really, really impressive. Changed the world impressive. What's up with this one? There's a weakness. What is it saying? Let's basically, what is the conclusion?
Cats are susceptible to COVID.
Okay. How did they get to that conclusion? Do a basic. How did they get to? I don't need details. How did they get to that? What did they do basically?
kill a bunch of cats?
What's a bunch?
I had no idea.
You should check that, right? What's a bunch?
They only use 3 cats.
They only use 3 cats. They use 3 cats. They use, I think, 3 ferrets, 2 dogs, one or 2 dogs. How do you run statistical analysis on an N of 2 or 3? How do you do that? You can't really, which is why they didn't do any statistical analysis. They pretty much just gave us raw data like a middle school kid. No, I mean, really, right? I mean, that's what when I talk about statistics with you guys in AP or honors, I talk about the problem that I run into as a judge for state science fairs is these middle school kids are like, I measured this and it was different. How do you know it's different? How do you know it's real? Well, the way statistics does it is it looks at a large sample, a powerful sample, let's put it that way, a sample that has enough power, enough size to be able to tell us something true. And it speaks for the whole population. Does 2 cats, 3 cats, how many cats? 3? Does 3 cats, is that enough to speak for the whole population? I mean, I don't think that this shouldn't have gotten published. I think it's timely, right? When we're in the middle of the biggest medical crisis in the last 100 years, getting out papers like this are really important. Do you publish them in science? So here's my question, Mikey. Do you publish it in science? Do you think this was worthy of science?
I think that if there was more data, then it should be, but as it stands right now, I do not.
Anybody else have a comment? Do you find these journal clubs to be torture or are they worthwhile? I need some feedback. These are
This one was kind of boring because like the implications weren't really like towards humans.
Yeah, but so a lot of you guys will interpret it, I think, the way that Mikey did, which was that this has implications for testing vaccines for humankind, right? And that's fine. That's a fine interpretation. You could look at it another way and ask, well, what does it tell us about actual physiology that's normal? What does diseased physiology, so pathophysiology, actually tell you about its opposite? About my phone is ringing. I don't know who that is. What does it tell you about its opposite? About what normally working functional physiological systems are like? In this, I don't get any of that. I haven't learned anything new about how physiology works. I've only learned that I should be scared of cats and ferrets because they can actually serve as a go-between, an intermediate species for infection. Like when you take your cats out and walk them at the cat park, if they mingle with other cats, they could spread this. You cat people, that's what you do, right? You like to put them on a leash and walk them out to... Somebody keeps calling me. Nothing? I got nothing from you. Oh my goodness. I can't wait till we're back in school where we can actually meet, because then I can like stare at you personally. And you'll have to...
Wouldn't this be a wonderful article if they actually had good results with actual statistics to help disprove the underlying foundation of PETA, perhaps? Maybe.
It's kind of a political message, maybe. Though we try to, as scientists, we shy away from doing that because it could also, I hadn't thought about this before until you said that, but what about the anti-vax movement? If you pair this with a vaccine showing the vaccine knocks down how contagious they are, what their viral load is, that's good information too, right? It makes it a stronger paper. All this said was that they carry the virus. It's not that cool. It's not that cool. So I think even when you look at it from all those other angles, I think Marco's probably right. Yeah. All right. Well, next week, please prepare yourself for to ask questions. Orthopedic surgeon, I believe, according to her bio, she specializes in women's orthopedic medicine because contrary to woke opinion, females and males actually do have slightly different physiology. For example, females do tend to tear their ACLs at a higher rate than males do. So if you're thinking about any type of medicine, have your questions ready.